Okay, so it's my great pleasure to introduce Shihui. He's a real pioneer in cultural neuroscience, and he's from Peking University. So Shihui.
Okay, thank you very much. I appreciate very much the organizers and kind invitation for me to this. But I think it's a very exciting symposium. Last 1.5 days, people are talking about culture effect on human cognition, human brain activities, and also talking about genetic effect, and also about gene-culture interaction on human cognition. I think the topic is very exciting. But I think there's still one issue which really, I think it's a very important issue, but has not been really, it's difficult to find a proper method to address this issue. That is how gene and cultural interaction influence the human brain activity involved in multiple cognitive processes. So today I'm going to present a short study that tries to explore a possible way to address this issue. So I focused on the one candidate gene, that is the serotonin transporter gene, how this gene will moderate the association between cultural value and the social brain network involved in social cognition. So social cognition is basically it consists of two components. One is the process of self, and the other is the process of the other. So I believe that I'm a professor, so that's why I come to this conference. And you, because I believe you might be interested in this topic, that's why I gave a talk with this topic. I think social cognition is really critical for people, human behaviors and social communications. So in terms of the self-concept, I think this popular theory proposed by Professor Marcus and Kiyama almost 20 years ago. And according to this theory, there's a strong cultural difference in self-concept. The self-concept in Western cultures is a bounded structure with unique dispositions or traits of self that keep invariant across different social cultural contexts. But the self-concept in East Asian culture is a variable structure, and that stresses the fundamental connections between the self and others. So if you look at the figure in these models, so in the Western cultures, the self has a solid boundary, and there's no overlap between self and others, even close others like friends and parents. But for the self-concept in East Asian cultures, you see the boundary is not that solid, and also there's a strong overlap, some kind of overlap between the self and close others, such as mother, father, and best friend. I want to give you an example to illustrate the cultural differences in self-concept. If we show subjects her own face and with another familiar face, like a friend face, classmate, or roommate, and ask this subject to identify the orientation of this face, toward the left or toward the right, and we measure reaction time. What basic finding for such a study is that for most of these college students, either Chinese students or American students, they responded faster to their own face compared to their friend or face. So this is what we call the self-face advantage in reaction times. However, such a self-face advantage can be easily influenced by social context in Chinese subjects, but not in American subjects. What we did in these two studies is that we show subjects her own face and her supervisor's face, or another faculty professor from the same department. For Chinese subjects, they still respond faster to their own face compared to another faculty's face. But when they watch their own face and the supervisor's face, they respond much slower to their own face compared to their supervisor's face. Seems that Chinese students are really scared by their supervisors. And my students call this a boss effect. So how possible such a boss effect can also occur in Americans? So we did this similar experiment on the students from Southern California, and we see in this figure they respond faster to their own face compared to faculty face. They also respond faster to their own face compared to their supervisor's face. So American students are not scared by their supervisors. So they are much more likely, I think, than Chinese students are. So this is a very simple example. This is how self-concept can be influenced by social context, the social experiences. I think during the last 10 years, research in the field of neuroimaging tried to explore the neural subjects of self-concept, self-reflection. And there has been increasing evidence that the neural subjects underlying self-concept are strongly modulated, influenced by culture and also by sensory experiences. But also, a few studies talk about the genetic effect on the neural subjects underlying self-concept. One paradigm used in this field is the self-referential task. The task is quite easy. Subjects were shown these personality trait objectives, and they have to make judgment whether a trait can describe itself or can describe a public person. So they used George Bush in this study. The basic finding is that when consciousing, comparing to this public person judgment, self-judgment induces much stronger activity in the medial prefrontal cortex. So that's the brain area that has been associated with self-reflection. In terms of the cultural difference in self-concept, like this model, so we thought about, given that there's a cultural difference in the self-concept, like this model show, whether it's possible, there might be a can we give a neuroscience account of such a cultural difference in self-concept? It's possible that for individuals in Western cultures, the brain, the neural representation of the self and other are also separated from each other. So to correspond to this model of self-concept. But for Chinese, like people from East Asian cultures, the neural representation of the self and mother might be overlapped. So in corresponds to this model of the self-concept in East Asian cultures. So to test this idea, we scanned both Chinese students and English-speaking Westerners, who also are college students studying in Beijing. So the basic finding is that for both cultural groups, when they thought about the self, we observed brain activation in the medial prefrontal cortex in both cultural groups. However, when the subjects thought about their mother, how smart their mother is, answer such questions, Chinese subjects also show activation in the same brain area, the medial prefrontal cortex, when they think about their mother. But this is only true for Chinese subjects, but not the Westerner students. It seems that these neuroscience findings support that at the neural level, there's also overlap of the self and the close others in Chinese subjects. But in Westerners, the seems that self is isolated from mother, even close other. And one of our recent studies, we try to more directly examine the cultural influence on the neural representation of the self. So now we scan Chinese and Danish participants. When they reflect on either the physical attributes or the mental attributes or social roles of the self and public person. So in this study, what we found is that relative to Chinese subjects, you see Danish subjects show much stronger activity in the medial prefrontal cortex when they think about the social role, the mental attributes, or the physical features of the self, compared to what we observe in Chinese subject speakers. So this activation is much stronger than what we observe in Chinese subjects. But in contrast, the Chinese subjects show activation in the TPG, the temporal parietal junction, that has been associated with theory of mind tasks, but only when Chinese subjects reflect on their social attributes. I'm a professor, I'm a student. When they reflect on their social roles. It seems that when Chinese subjects think about their social role, they might take others' perspective using this so-called theory of mind area. In addition, when we examine the correlation between the brain activity involved in self-reflection and measure of cultural value, the interdependence of self-concept, we found that the brain activity involved in self-reflection is correlated with this measure of cultural value. So here we show the positive correlation for the TPG activity, but there's a negative correlation for the MPFC activity. So these data suggest that there's a kind of association between the brain activity involved in self-reflection and the culture, one cultural value. So from these whole subject group. It seems that I think these data suggest that the people from different cultural contexts, from East Asian cultural contexts, from Western cultural contexts, might use distinct neural strategies by increasing the weight, either like the MPFC or TPG, these different brain regions during self-reflection. So the study I'm going to talk about, I think sort of a brief, very short study. We try to examine, given that we found an association between the brain activity involved in self-reflection and the interdependent cultural value, how such association can be influenced by a specific gene. So that's the question we want to address in this study. We selected a candidate gene, this serotonin transporter gene. So this slide, I think Dr. Jun showed this slide yesterday. The short and long allele subjects are characterized by the differences in the concentration of serotonin in the synaptic cleft. That's the so far literature told us about the differences between short and long alleles. So why we select this candidate gene? There's a couple of reasons. First of all, depression symptoms and neuroticisms are more sensitive to life events in short than long allele carriers. So these are the basic findings we report from behavior studies. And also, I think yesterday Dr. Rincha also showed one of her studies suggests that the frequency of this serotonin transporter varies across nations with different collective cultural values. It seems that the more a nation with a stronger collectivist cultural values, the proportion of short allele carriers is much larger. So if you remember that positive correlation between the culture. So that's the reason that we select this gene. So we hypothesized that these serotonin transporter polymorphisms may interact with the interdependence of self-construct, the cultural value, to shape the social brain network related to thinking about self and the close other. So that's our hypothesis. So how to test this hypothesis? So again, we use a self-reflection task or self-reflection processing task. We show subjects 80 personality trait words, either positive or negative. Ask them to make judgment whether such a trait words can describe the self or their mother or another public person. And also we have 80 items to describe people's physical features, like the color of the hair, how long you have a long arm, small hand, such a physical body features. And also 80 items to describe the social roles of the person. So we show these items in separate blocks of trials and ask subjects to make judgment on either the self or their mother, a close other, or public person. So, in this study we have only Chinese subjects and 17 short allele carriers and 17 long allele carriers. You know that because the proportion of this long allele carriers are very small in Chinese sample, So we have to get the blood sample from almost 300 subjects, and to get I think around 25 long allele carriers. So for these imaging studies, so that's why I think there's a limitation for imaging studies to have a large sample of the genotype subjects. So then we use this Singalis construal scale to measure the cultural value, the so-called interdependence. In this study, we use the difference between interdependence and the independent items to get one value as index of the cultural value. So for the data analysis, first of all, we run a simple whole brain regression analysis to across all these two genotype groups. There are 34 subjects. So the first step of regression analysis will show the brain regions that are associated with the cultural value. To show the association between the brain activity involved in either self-reflection or reflection on mother and with the measure of cultural value. And then the second step analysis, we conduct a multiple regression analysis and use the measure of independence and genotype as variables. So this analysis will let us know in which region the brain activity, the association between the brain activity and the cultural value are moderated by this genotype. So that's basically how we analyze the data. I want only two slides to show the results. The first slide shows the moderator effect of this gene on the association between interdependence, this cultural value, and the brain activity underlying self-judgment on mental attributes, on their personality traits. If you look at the figure, so basically what we found is that in some of these brain areas, like the middle frontal cortex, the precuneus and TBJ, and the middle temporal cortex. So in these brain areas, the activity associated with self-reflection on mental attributes are correlated with the measure of cultural value, interdependence. There's a negative correlation. But this correlation was only evident for the long allele carriers. So there's no such correlation for the S allele carriers. In other areas, like the medial prefrontal cortex, the inferior frontal cortex, the insular, and the hippocampus, we also found correlation for the L allele carriers. Now here it is a positive correlation between the brain activity and this measure of cultural value, but no such correlation for the S allele carriers. So seems that this result show a kind of evidence for how this gene, this serotonin transporter gene, can moderate the association between a cultural value and the brain activity involved in self-reflection. The next slide shows the results for the genotype effect on the association between the interdependence and the brain activity underlying mother judgments on personality trait. Again, for this slide shows that the correlation between the brain activity and the cultural value was also only evident for the long allele carriers. See, this is the results in the precuneus, in the insular, in the MPRC, and in the right frontal cortex. So this correlation was observed only for L allele carriers, but not for S allele carriers. So these are the main results that we found in this simple study. So to give us a short summary, so first of all, I think our data show evidence for the association between cultural value and the social brain network activity during self-reflection. And these actually are related to self-representation. I think these results are not something new, because previous papers also reported such an association between cultural value and brain activity involved in social reflection. But the new results I reported here is that we found that such association are strongly modulated by this gene, this serotonin transporter gene. To make a simple conclusion of this study, I think culture, this gene-culture interaction theory is becoming popular and hot now, but there has been few evidence to reveal how the interaction between gene culture can really shape the brain activity involved in human cognition. To give a brief summary of the previous studies, I think so far our study has shown evidence that the culture influences neural repetition of the cell. We show that these culture group differences, and also show that the variation of the brain activity involving self-reflection across the one culture group, or across individuals in one culture group. We also show evidence that temporary priming of subjects by culture subject can also modulate the neural substrates involved in self-reflection. And also we have shown that sensory experiences also affect the neural repetition of the self. I have no time to tell the story of this study. But what I want to show here is that there's kind of a preliminary evidence for the gene-culture interaction on the brain activity involved in reflection of the self and the closed other. So all these studies are put together indicate that I think the question is what makes the self? How people think about the self? All these data suggest that the neural repetition of the self can be influenced by either sensory experiences and strongly influenced by cultural experiences. But at the end of it, I think the brain is a biological brain. It's a biological organ. It is some degree should be influenced, determined by the biological factors such as gene. So what we show here is that the gene might modulate the association between the cultural values and the brain activity involved in self-concept. So that's the study I want to report here. And finally, I just only one slide to let you know that I think because this field is developing rapidly, and we recently launched a new journal with a title called "The Culture and Brain" with Springer publisher. I think this journal, the target of this journal is to provide a platform for people, at least for people in this room, to publish your studies and so that we can promote this interdisciplinary communication exchange for the future development of this field. Thank you very much.